Hello everyone, welcome back. We are on day 2 of our 90 days bioinformatics challenge. Today we are looking into the biological databases, essential tools for bioinformatics. We will try to understand how databases are classified and identify the key examples. Let's get started. Let's rewind to the early days of bioinformatics, a time before supercomputers and automated pipelines. How it all began with Margaret Dayhoff, the pioneer of bioinformatics. Margaret Dayhoff was an American physical chemist who made groundbreaking contributions to bioinformatics in the 1960s. She is best known for creating the first biological database, the Atlas of Protein Sequences and Structures, which was compiled with 1573 protein sequences, and now this is PIR, Protein Information Resource. Also, she devised the single letter codes for each amino acid that are widely used today in bioinformatics. We'll look into this in upcoming sessions. Database allows us to store, retrieve, and analyze the ever-growing flood of biological data. And now coming to the types of databases, based on the source of information, they are broadly classified as primary and secondary database. Primary database are the one which consists of raw unprocessed data submitted by researchers. Think of primary database as the first stop for biological data. Secondary database are derived from primary databases with added interpretation and annotations. These databases are like value-added services helping us make sense of raw data. So now, based on the type of data they are stored, the databases are classified as sequence database that holds sequences, structured database that holds 3D structural informations, and specialized databases that holds focused informations. Coming to the sequence databases, primary database, these are the repositories of raw nucleotides or amino acid sequences that are directly deposited by researchers. Example for this are GenBank hosted by NCBI, USA, EMBL hosted by European Bioinformatics Institute, Europe, and DDBJ hosted by NIG, National Institute of Genetics, Japan. Secondary databases that holds functional annotations that are derived by fetching the sequence from primary database. Examples are UniProt that provides functional insights into the proteins, and PFAM that provides conserved protein domain family informations. Next, coming to the structural databases, PDB that provides the experimentally derived 3D structure informations of proteins and nucleic acids. Under secondary database, we have SCOP and CATH that provides SCOP provides structures that are classified based on families, and CATH provides structures classified based on structure and function. Coming to the specialized databases, these are focused on specific organism systems and biological processes. Under this, KEGG, Kite Encyclopedia of Genes and Genomes, provides metabolic pathway and gene functions, and OMIM provides genetic disorders and associated genes, and ClinVar provides clinically significant genetic variations. So this is all about specified databases. So now if you want to know how many databases are existing and what kind of informations are provided, you can access the informations of all the databases that are available by visiting the Nucleic Acid Research Journal database issues that is published every year with the recently added database informations. So this is one of the database providing the 97 new databases were added last year and summing up to overall 1959 databases. And that's it all for today. And just to rewind, biological database are the information of bioinformatics research that are classified into primary database that provides raw data, secondary database that offers significant insights, and specialized database that focus on specific topics. So that's a wrap up for today. Let's explore these databases together in future sessions, and comment below which database you are using most and which database you got to know and what new info you got from this. Thank you for joining me on this 90-day bioinformatics challenge. See you tomorrow. Until then, bye-bye from Jnanendra Shanmugam.